Well, thank you for that really appropriate introduction for this, what I'm going to talk about, which is intratumor heterogeneity. It is a pleasure to be back at Mount Sinai and in New York during, I think, the best time of the year, and to see all of you guys again. Thank you to Fluidigm for the invitation to come talk about my work at this event and for putting this on. I think Adib was saying to me earlier today that this is still such a small community that it's important for all of us to really get to know each other. I think that's a great point, that the speed at which things are advancing is so fast that publications are, if you wait for the publications to come out, you're going to be behind the game. So I think this is a chance for all of us to really network and keep in touch and share our knowledge about this new platform. So I'm going to use the mouse as my pointer so the people on the stream can see this. And okay, so I'm going to talk about actually acute myeloid leukemia as my model of intratumor heterogeneity. This is a topic that I've been obsessed with since 2006 or 7 when I started my PhD work with Gary Nolan. And I am fascinated by this concept that cancer has so many different cell types within it. And we're starting to apply this now to glioblastoma in my postdoc work at the UCSF. But the work you're going to see here was done in Gary Nolan's lab at Stanford. So briefly, I work for Fluidigm as a consultant, and I own stock in Fluidigm. So I'll try not to be too enthusiastic about the platform, but let the science speak for itself. So cancer, as Dr. Birkhoff introduced, is a heterogeneous disease. There's intertumoral heterogeneity, which is the difference between person to person. The cancer that one person gets is not identical to the cancer that another person gets, and we're very familiar with this concept. More and more now, I think we're starting to appreciate the importance of intratumoral heterogeneity, which is the observation that in solid tumors and leukemias, there are many different cell types within the tumor that all share a clonal origin with the same initiating mutations. They may acquire subsequent mutations that make them different. So this is a slide of glioblastoma where you can see several different cell types within the tumor. And these can be immune infiltrate, these can be epigenetically distinct cells that are actually part of the same clone but have taken on slightly different phenotypes in response to their environment or just due to stochasticity. And these can be truly genetically distinct subclones as well. And this is probably the most important one in terms of therapeutic resistance, because we see now in lots of studies where patients are given kinase inhibitors, targeted inhibitors for cancer, we see these escape mutations basically being evolutionarily selected in subclones that have a lucky mutation that allows them to escape that inhibitor. So all three of these contribute to what we see as cancer heterogeneity, and we need platforms, single-cell platforms, that can really get at this. So I'm going to tell you three brief stories. The first one is published, but I just want to introduce this analysis approach called visne, and I'll show you how we use that to pull out rare leukemic cells in AML and ALL. The next story is about pediatric AML, where I used CyTOF as sort of a looking glass at the total amount of heterogeneity in the cancer to know exactly where we should pinpoint our efforts for subsequent genomic investigation. I'll show you how that worked in a moment. And then finally, a longer story that's also unpublished, working with collaborators at Columbia, where we're using CyTOF to get a feel for the landscape of different phenotypes in pediatric AML. And the very exciting result of this work was that we've pulled out what we call a signaling-based definition of primitive cells in leukemia. So this is the published work that I'll just get through very briefly. This was published about a year and a half ago in Nature Biotech, and this was also a collaboration with our friends at Columbia, Donna Pear, and her student Elad Amir. The algorithm here is called visne, which is based on an algorithm published several years ago called tsne. And what visne does is it sort of organizes your single-cell data. The challenge here is, as I'm sure many of you are aware, is that CyTOF gives us very high-dimensional data of a degree that we've never really had to deal with before. There's no other platform where you have to deal with millions of events, millions of observations in 40-parameter space. And this is a good thing because it allows us to get at biology we couldn't get at before, but it's also a challenge analytically. So what visne tries to do is similar to principal component analysis. It tries to compress that 30 or 40 parameter information down into a two-dimensional plot that you can interpret, because humans have a really hard time interpreting anything greater than 3 dimensions. So if you have this fictional data set here, which is just pretend single-cell data in three-dimensional space, you can see there that it sort of has this ribbon shape to it. And there's this region in cyan and this other region in magenta that are closer to each other in Euclidean space. Most dimensionality reduction algorithms, taking this three-dimensional data and compressing it down to two, will put those two next to each other. And the reason we like visne is because it actually follows the contours of the data. It pays more attention to what's called the local geometry, which is those bumps and dips in the data as it follows along that ribbon. Rather than putting the cyan and magenta pieces next to each other, it appreciates that there's this continuity to the ribbon here. And we see these types of shapes in the data in biological developmental systems. So these show up a lot in CyTOF data, and it's really nice to have an algorithm that can display them for you in an intuitive way on an XY plot. So we've applied this to many different systems now, and I think you'll see several different ways of using this in the rest of my talk and also in other talks today. Here's a couple quick examples from the paper how we use this to look at leukemia. So in this first example, we have AML diagnosis and relapse bone marrow, which we integrate into the same data file. So these were measured separately. They're from the same patient, and we measured them separately and integrated them in the same data file and run visne on that file. And you get this plot where the single cells, those little dots, are spread across this sort of horseshoe shape. You have a population out here on the left, which is mature lymphoid cells, and up here, which is other mature erythroid cells. But most of the leukemia is restricted to this horseshoe shape. And what's interesting, you can appreciate right away, is that the purple cells here are the diagnosis cells, and the red cells are the relapse. So there's a phenotypic shift that's happening from diagnosis to relapse. As we look at the individual markers from those cells, I think you'll appreciate sort of how the algorithm is organizing the data on this plot. CD5, of course, is a marker on T cells and B cells. CD7 is on T cells and NK cells. So you see that there's these mature lymphoid cells out here on the left, but you see a population of CD7 positive leukemia cells. So the algorithm's integrating information from all of the markers that we measured rather than just prioritizing one or the other. And this is something that humans can't really do, so it's really nice to have an algorithm that can do that. As I flip through some more markers, you can appreciate how there's a very primitive side of the leukemia here, which is CD34 positive. And for those of you who don't work on AML, I'll just mention that CD34 is a very important marker in sort of all blood diseases, and it's a primitive marker of normal blood cells. And we look at it in AML to sort of get a feel for the most primitive cells in the leukemia. And this will come up a lot later in my talk. So there's this primitive half of the leukemia here, which is enriched at relapse. And as I flip through the rest of the markers, you can see all of the heterogeneity within that leukemia. Now at relapse, if we focus on only those cells, you see they are largely out here on the right side of the plot. But what we found fascinating is that at diagnosis, there are these rare cells in here in purple, which recapitulate the phenotype of that relapse clone. So even though these two samples were acquired a year apart, in terms of the clinical timeline of this patient, the diagnosis clone, or the diagnosis sample, has cells that resemble the relapsed leukemia. And so we have not yet done the experiment, but the appropriate experiment here is to actually sort out those cells at diagnosis and see if they have molecular features that resemble the relapse clone. And my second story now, I'll show you a little bit of that type of work. Now the final example here to explain visne is pediatric ALL. I'm just going to flip through this very quickly, but the general idea here is that you can use visne to get down to very rare cell types. So here we mixed healthy bone marrow with pediatric ALL bone marrow, which we labeled with a special metal label so that we could identify it later in the analysis. So this was actually a synthetic sample that resembles what's called minimal residual disease, which is the phenomenon where after treatment with chemotherapy, there's residual leukemic cells remaining in the bone marrow from most patients with AML and some patients with ALL. So we made this synthetic MRD sample here, and we applied visne to that sample, and you can see that there's healthy bone marrow here in blue and synthetic MRD marrow here in purple. So what we're really looking for is any phenotype that's unique to the synthetic MRD sample. Those would be the cells that are basically leukemic blasts and have a phenotype that's not observed in the normal marrow. So if we zoom in on this little island down here, we find that it's almost entirely purple cells. And sure enough, when we decode the data and pull out which cells we labeled in red here with our special metal barcode, we find that all of the leukemic blasts are restricted to that island, with the exception of some normal cells that were in that leukemic bone marrow. And if we zoom in on the phenotype of that little island of leukemic cells, we find that they are 34 positive, CD10 positive, 45 low, and 15 mid, which is the phenotype of the leukemia that we observed. So this is a way to take a lot of cells, this was about 10,000 cells, and drill down and find a very rare population. Okay, so I just wanted to give you that familiarity, I hope, with visne, because you're going to see that used over and over in the subsequent stories. This next story is unpublished. This is a collaboration with St. Jude and Jim Downing's lab, and Amanda Gedman, who was a postdoc there, and our friends at Columbia again, Donna Pear, and a grad student of hers, Jacob Levine, and my colleagues in the Nolan lab, Sean Bendel and Kara Davis. And what we're interested in here is really the difference between those two different types of heterogeneity that I introduced at the beginning, genetic drivers of heterogeneity and epigenetic drivers of heterogeneity. And we see this heterogeneity, similar to what I showed in the last slide. We see that there's a lot of different phenotypes in AML, but we can't really describe where it comes from. Is it coming from the genome, or is it coming from the epigenome? And AML, for those of you who don't work on this, is thought of as a developmental hierarchy. Similar to the developing blood, there's a lot of evidence that in AML you have primitive cells that mimic the stem cells, the most primitive cells in your bone marrow. And the classic model, and there's evidence now for this in genomics work, is that you need a genetic mutation that impairs the differentiation of those cells and allows them to maintain a stem cell fate. And then you need an additional hit that causes them to grow out of control. And that will give you your very first leukemic stem cell. Now that cell can go on and acquire additional mutations, but it should be able to create the initial leukemic clone just by itself. Now those additional mutations could occur in that initial pool here or in the leukemic stem cell, and then you'll get a competing subclone that's competing for space in the bone marrow. And there's a lot of next-gen sequencing data now that supports this model. So what we were interested to know is, in this particular patient that we have, which was very unique, are there second hit mutations that are unique to the different subclones? And do those different subclones have the same phenotypes by CyTOF? So this patient, where we could ask this really unique question, was a sample that was given to us by our colleagues at St. Jude, and they had observed in their next-gen sequencing data that this patient had mutations in NRAS, and it had two different mutations in almost adjacent amino acids. So it had G12G and G13D mutations in two different subclones. And so you could see in the sequencing reads that these were two very distinct subclones, and we could see from the frequency of these different sequencing reads shown here that the frequency of those subclones is dominated by the NRAS G13D clone here in blue, and then there was a minor clone that had the NRAS G12D. So both of these were coexisting at diagnosis in this patient. There's also healthy contaminating cells which sort of bring the overall allele frequencies down below the expected 50% here total. So we took this patient sample, analyzed it by CyTOF, looking at 31 surface markers for any clue as to where the phenotypic heterogeneity would be in this sample. And we pulled out, similar to the plots that I showed you before, many different mature cell populations here around the periphery, and this larger continent of leukemic and myeloid cells here in the middle. Similar to the last sample I showed you, there were about half of the cells were 34 positive here on the left, and then other markers that I'll use, HLA-DR, CD117, and CD64, sort of break up the rest of this heterogeneity. So our question was, where are those different subclones? We know there are two different subclones, and the very basic hypothesis might be that the minor subclone is over here on the right and the major subclone is over here on the left, but we wanted to figure this out. And this really gets down to the core question, which is this heterogeneity that we're observing here, all these different cell types, is this driven by epigenetics or genetics? Because if we find that there are genetically distinct cells distributed across this phenotypic space, then that's more of a genetic influence. But if the two subclones are evenly distributed across this phenotypic space, then all of these phenotypic differences we're seeing are really driven by non-genetic mechanisms. So we did the experiment. We sort of chopped up this continent of leukemic cells using fluorescent flow cytometry, using an 8-marker panel that was determined based on our analysis of this CyTOF data. So we picked out the best 8 markers, we repeated the analysis on the same sample by flow, and we did the visne analysis on that data. And so you get a similar plot where we have the same overall features, that there's a 34 positive half, HLA-DR high, 117 high on the left here, and 64 high on the right. And so this allows us then to do sorting, where we can cut out different pieces of the phenotypic diversity in this patient, sort them out into different tubes. So we sorted out 7 different populations within the leukemia. We sorted out T and B cells as a control, and then subjected these to ultra-deep sequencing, where we sort of already knew what we were looking for. We knew from the whole genome sequencing of this patient about 350 tumor-specific variants, and so we were able to do ultra-deep sequencing where we selected, where we enriched the DNA for those particular variants. This allowed us to get down to 2,000-fold read depth on each of those 350 variants. And with that type of data, you can look at how the allele frequencies shift across the different subpopulations in the tumor. These are phenotypically distinct populations, called 2 and 4 here, based on the previous plot. And what we find is that these 350 tumor-specific variants, shown here as dots on the plot, many times they correlate. That suggests that the subclones are present at the same frequency in these different populations. But we can also find populations where, such as population 1 here, where there's an enrichment of a group of these different mutations. And so we can use this information to pull out the clonal family tree, really, of this leukemia, where we can assign mutations, those tumor-specific variants, to different subclones, which we call A1, A2, B1, and B2, because there's evidence from which ones are inherited that these A1 and B1 clones were the initial clones, and then there are granddaughter clones, really, of each of those. And all of these are competing for space in the leukemic bone marrow. So what we found is that if we look at the different phenotypes in this space of CD117 by HLA-DR by CD34, there's this sort of continuity, this path, this developmental path that sort of goes through this CD34 and HLA-DR space. And if we align the data based on that, we can order those different populations from most primitive to most mature, where the most primitive ones have high expression of 34 and low expression of 38 and low expression of HLA-DR and 117. And if we order them by that, we can then look at their frequency. We find that the most abundant phenotypes in our data were populations 4 and 5 here. The healthy sample as a control, the healthy cells in the same sample, this is using the genetic data of the non-mutated alleles at those 350 sites. We were able to quantify the abundance of healthy cells in each of those phenotypes, and we found that the healthy cells follow the expected trajectory, that the most primitive cells are quite rare, and as the blood system grows, you see hematopoiesis, and then you see expansion of the progenitors and the more mature phenotypes at the end here. And now we can get to, I think, which is the grand finale of this data set, which is the abundance of each phenotype broken down by subclone. So again, just to orient you, each row here is a different phenotype that we observed, 7 different phenotypes in the leukemia, from most primitive to most mature. And then this is the family tree here, with A1 and B1 as the daughter clones and A2 and B2 as the granddaughters. And what we find is that B1 and B2 are the most abundant clones. This is by far the most successful clone in this leukemia. Both of them have this NRAS G13D mutation. We find very rare cells that have the parental mutations in ALAS2 and FOXN1. And then over here in clone A1, there's a really fascinating observation that there is an enrichment of clone A1 in this most primitive fraction, but then it fails to differentiate down and acquire these additional markers. And we think that this is driven by probably senescence in response to this very powerful oncogene NRAS G12D. And it's not until it acquires additional mutations, such as this RAC2 mutation, that it's able to escape that primitive fate and differentiate down the line. And then it's basically phenocopying the clones B1 and B2. So this is an evolutionary strategy of 3 different subclones that we're seeing here, B1, B2, and A2, that have managed to figure out how to occupy all these different phenotypic fates and become successful clones. The overall takeaway here is that the epigenetic program in those clones is conserved. They're all occupying the same phenotypes, but that second hit downstream of that, the initial hit that sort of locks them into a stem cell-like program has to be just right. The G13D seems to have been the most advantageous hit for this leukemia, and G12D was just a little too strong until it acquired subsequent mutations. And then finally, the phenotypic heterogeneity that we see here is mostly driven by epigenetics. We did not see distinct phenotypes across the different subclones. So now finally, I'd like to bring you through a really cool story where we're using CyTOF to look at the entire phenotypic landscape of pediatric AML. So in addition to that one patient that we just analyzed, we did a very similar type of analysis on 15 additional AML patients and 5 healthy controls. And this was with the same group of people that I just mentioned in the last story. And what we're looking at here is the signaling biology in hematopoietic stem and progenitor cells, and how the leukemia sort of mimics or basically phenocopies that signaling biology that we see in normal primitive stem cells. So this was an observation out of Gary's lab in 2011 that hematopoietic stem cells in healthy bone marrow have distinct signaling biology. Kenny Gibbs showed that they respond to GCSF through STAT3 and STAT5 very strongly, and as they differentiate into more mature cells, they begin to lose that STAT5 response. And ultimately, as these GMP and MEP progenitors, committed progenitors, they've lost that response. And then finally, when they become true monocytes, they regain the GCSF STAT3 signaling node. And this sort of explains an observation 7 years prior from 2004, where Jonathan Irish showed that in adult AML, if you look with conventional flow and stimulate with GCSF, just as before, if you look at the number of AML cells that respond to the GCSF through STAT3 and STAT5, both of them, that was the primitive phenotype that we just talked about. That observation is correlated with the clinical outcome of these patients, that if they activate both STAT3 and STAT5, those patients do poorly, but if they only activate STAT3, they're sort of in the middle, and if they don't activate either very strongly, then they do well. So this was a very interesting observation, but it was really difficult to explain, I think, until we started to understand how this was rooted in normal stem cell biology. So what Sean and I did with our pediatric AML cohort from St. Jude is we analyzed these 16 diagnosis AML bone marrow samples and 5 adult healthy bone marrow samples with a panel of 17 stimulation conditions. These are largely cytokines and chemicals that activate signaling inside those cells. So we can really see what are the signaling pathways within those cells, what are the behaviors, what is the wiring in each of those leukemic cells. And then we break up the heterogeneity within each leukemia using these 16 surface markers down here. And we observe the outcome of those perturbations on these 15 functional markers up top. So with this 31 marker panel, CyTOF panel, we acquired millions of data points across our cohort, an average of 200,000 data points per patient. And we were able to use this new algorithm that is not yet published and will be co-published with this data called Phenograph, which is developed by Jacob Levine, a grad student in Donna Pear's lab. And what this algorithm tries to do is it's essentially a clustering algorithm, but the existing clustering algorithms weren't working very well in this massive data set. So this is a very fast and very smart, I think, clustering algorithm that looks at the data as a graph. And just like you could make a graph of everybody's relationship on Facebook, you could graph who's friends with whom on Facebook, and you would end up with these subpopulations where you see densely interconnected clusters of everybody that went to the same university, or everybody that lives in the same country, or everybody that works in the same type of science, let's say. And you see these communities within the graph of everybody interconnected on Facebook. You see the same thing here, that the cells that are most similar to each other and closest to each other in high dimensional space are linked with many interconnections, and the algorithm is able to identify those communities, which you can sort of see here in this plot. They're driven by the surface marker expression, and the algorithm automatically and in an unsupervised way assigns each of these cells to a different community, which is a different type of phenotypes. And so we get an average of 20 or 30 phenotypes per patient. And within each of these phenotype communities or subpopulations, we see the intracellular signaling behavior of all of those stimuli by all of those phosphomarkers that we measured. We get a total of 224 signaling behaviors that we can use to look at the wiring in each of those communities. At the phenotype level, here's how that data breaks up. So this is the visne algorithm, again, using the t-SNE algorithm to give you an overall view of how these different phenotypes relate to each other. So if we just do t-SNE on the surface markers that we measured in this data, each of these little colored dots is a different cluster from a different patient. The size tells you the number of cells inside the cluster, and all of the healthy patients are grouped here in bright blue. So you can see that most of the healthy cells predominate down here at the bottom of the plot, and there are some rare ones up here at the top of the plot. When we look at the surface markers on those nodes, it starts to make sense that the top of the plot is more primitive and expresses CD34. There's CD117 on the right, 123 here in the middle, and then HLA-DR really scattered all over the plot, and then finally CD64, a very mature marker, a marker of monocytes down at the bottom. The very simple way to think about this is that it's really just oriented from primitive to mature, from top to the bottom of the plot. This gives you an idea of the total phenotypic diversity that we see in our cohort, that there are patients who are scattered all throughout this plot, and then there are patients like this one here in green, which was restricted mostly to one phenotype and had a few random nodes in other phenotypes. Now what we wanted to use is that signaling biology. And first we had to describe the signaling phenotype of healthy stem cells and other healthy cell types in great detail. So using the information from those induced stimuli, you can see several different stim and phospho combinations down here, a subset of the 224. We looked at our 5 healthy samples and pulled out signaling phenotypes that were significantly predictive of these 4 different healthy cell types. Hematopoietic stem and progenitor cells, HSPCs, early B cells, T cells, and monocytes. And so each of these different cell types has a behavior that is related to that cell type. So we can then use that information to look at the leukemia. And the most important piece of information for the rest of the talk is this signaling phenotype that is specific to healthy HSPCs. So our question is this signaling phenotype that we see in HSPCs conserved in AML? In other words, is what Jonathan Irish observed in 2004 just the tip of the iceberg? Are leukemic cells really recapitulating this primitive cell signaling pattern? And so this is an early glimpse at the data that gave us a hint we were on the right track. If we try to organize the data based on signaling, if we look at only the healthy cell types here organized by signaling, each row here is a different cluster from the healthy samples. And the data is clustered based on the signaling information, not based on the surface information. And what we find is that the top branch of this tree here isolates a CD34 positive cluster of the healthy cells. In other words, if all you know is the signaling behavior, you can find the primitive cells in a healthy sample. But if you do the same analysis on AML, it's a mess. There's all of this sort of random assortment of many different phenotypes related to the same signaling. So the surface markers and signaling behaviors are decoupled in AML, but they're tightly linked in healthy. And if we quantify this with statistics, we can find that CD34 expression in healthy is highly correlated to this signaling behavior, whereas in AML, the amount of CD34 expression is really all over the map. And what these bars are is these gray boxes here on the left. So the top healthy box here in dark gray is this box here. So we break these up based on their signaling behavior, and we find that CD34 expression in AML is uncorrelated to the signaling behavior. So then what if we go all the way and use the signaling biology to classify the AML cells? And we use Phenograph a second time. Now we're using it to identify the nodes, the clusters within the AML that signal the same as the healthy cells. So this is I know this is getting sort of complicated. Let me reorient you to this plot. Basically, the dark dots here are healthy cell types that we observed the signaling, and we know exactly what the signaling is in those labeled observations. And we're using Phenograph now to find the AML cells that have a similar signaling behavior and classify those AML cells as the most similar healthy type based on the signaling, not based on surface. If we do that, we come up with subpopulations of AML cells in every patient that behaves like an HSPC, and then we can look at the phenotype of those cells. So here's some very basic CD34 by CD38 plots, and what we find is that there are patients, so in healthy control H2, we see a very canonical 34 positive, 38 positive and negative population here. And in SJ05, the leukemic cells that behave like an HSPC, they have the classic phenotype of a hematopoietic stem cell. They are 34 positive, 38 low. But in other patients, they have totally non-canonical phenotypes, that they can behave like an HSPC, but they can have a 34 negative phenotype. And we call these cells IFPCs, for inferred functional progenitor cells. So if we quantify the number of IFPCs per patient, they can be extremely rare, or they can be very abundant, occupying almost the entire patient sample. And we can use this variability. So this variability across our cohort in terms of the number of those IFPCs, those signaling-based classified primitive cells, we can use that variability to pull out gene expression signature. So by looking at the bulk gene expression data from each patient, we can find the genes that are most correlated with the number of IFPCs that are in the sample. And so what we have here is the IFPC frequency here in purple, and there are patients with a lot of IFPCs, and they have robust expression of these 49 genes in the signature here on the right. Oops, sorry. Now this signature is full of really interesting genes here. It has some overlap, about 20 genes overlap with previously published signatures in AML, but it's a unique and novel signature of these primitive cells, again using a signaling-based definition, not a surface-based definition. And these genes here in red I've highlighted just because these are expressed on the surface of the cell and could be used to potentially isolate these cells or used for targeted immunotherapy. So what we have here is a gene expression signature that we can then apply to much larger cohorts. So we've applied this gene expression signature looking for significant prediction of survival in several independent AML cohorts. Here we have a nice separation Kaplan-Meier curves where the patients with high expression of that IFPC signature do poorly. Here's a separate cohort, and then here's a third cohort, so this seems to be very reproducible. And then finally, we can look at a similar signature, which I haven't explained, where we came up with a signature that's surface-derived. That if we make the assumption that primitive AML cells have CD34 and low 45, we can come up with an alternative gene expression signature that should predict those cells. And that signaling-derived surface-derived primitive cell signature has no predictive power in addition to our signaling-derived signature. So really, the signaling-derived signature is much more powerful than a surface-based definition. So I hope I've shown you now how you could use CyTOF in several different ways to look at tumor heterogeneity, using leukemia as the model of tumor heterogeneity, but I know that myself and others are starting to do this now in solid tumors. We can use it to get down to very rare leukemic cells in situations like MRD. We can look at the relationship between phenotype and genotype and epigenetics, using CyTOF as a guide so we know where to focus our energy on the phenotypes that we observe. And then finally, you can really use it to pull out a landscape of different phenotypes in AML and get down to the signaling biology, which is really what's driving cancer. I think we get too caught up in what surface markers mean in cancer when it's really the intracellular behavior of these cells that's messed up. And we can't put too much weight on surface markers when we have an opportunity now to really get to the ways that cancer cells signal. I'd like to finally thank my PI for all of this work. This was my PhD mentor, Gary Nolan at Stanford. My colleagues, who I tried to mention as I went along, Sean, Kara, Nima, Robert, and Rachel are informatics gurus in the Nolan Lab and fantastic to work with. My PI at UCSF is Bill Weiss, and I have to thank him for letting me come out here and share this work with you. My colleagues at Columbia are fantastic to work with, and St. Jude, and my colleagues on the SPADE work in 2011, Sylvia Plevritis and Penn Chu. My funding here at the bottom. And then finally, I'd like to mention that if you've ever wondered why CyTOFs are orange, it's because they're all Giants fans. So thank you so much for your time and for having me here. Mary?
So when you look at MRD, I was wondering whether you've used barcoding to mixed, maybe instead of using the healthy bone marrow. You guys did that?
I ran through that very quickly. What we did was we barcoded the leukemic marrow.
Right, I understand. But is it necessary to add this healthy big? So can we imagine to have, I don't know, 15 patients' marrow?
That's a great idea. I think we need to expand the MRD investigation to
Right.
many healthy controls and many leukemias,
Yeah.
and look for commonalities in what the MRD phenotype looks like, but also use many different healthies to get a map of exactly what healthy marrow looks like, so that whenever you see an aberration from that map, then that must be leukemia. And people are doing that with conventional flow at Hematologics in Seattle, but I think we have an opportunity here to do that with CyTOF. Nobody's really doing that yet, except perhaps our later speaker, Brent Farrell, who's going to talk about some AML work as well.
Mm-hmm. Then, Eric, when you look at this clonal population, I mean, is it somehow one of the limitations of CyTOF is that you cannot really assess the same clone that you identify by CyTOF at the genetic level and clonogenic potential, and that somehow it's always correlative with, right?
Yes.
I mean, can you expand a little bit on this? Because all this correlation that you show, the P0 to P1, in fact, is a correlation, right? So you don't know that P0 gives rise to P1 because you cannot test it. And you never know exactly the genetic of these clones because you cannot test it since the cells are.
Yes, the cells are exploding, so
Yes.
we can't do it. But in that study, we used the CyTOF as a roadmap to design a flow cytometry experiment where we could sort out those clones. That's how we got the DNA from those different clones. So we can use CyTOF as a way to come up with really smart flow cytometry panels.
Mm-hmm.
I don't think that there's going to be a situation where you need all 30 parameters to sort out a population. If you can measure all 30, then you can come up with the 5 or 8 or 10 that allows you to get down to the populations you're looking for. And that's what we did here. And so, what we've done is we sorted out those cells and done a genetic investigation. We tried growing them in clonal assays to really do the experiment that you're describing, where we can prove that there are unique subclones that have these genotypes, but unfortunately they would not grow. So we're trying again with xenograft assays, but that's exactly the type of thing we're trying to do. I think the roadmap is there. It's just technological challenges. Yes?
So in the case of the IFPE, did you know whether these leukemias had a mutation in some sort of signaling pathway, and that's what was making them independent? And then I guess as a follow-up, if that is the answer, then could you sort of predict, based on what you would see there, a growth factor dependent cancer versus one that's kind of cell autonomous, which may be more relevant to maybe solid tumors?
No, that's a couple of great questions there. We had virtually no genetic information on these tumors. We only knew about 8 different commonly mutated genes, NRAS and KRAS and CBP alpha and things like that. A couple of the samples had been submitted for whole exome sequencing. But what we found is that there are dozens, if not hundreds, of mutations in each sample, and it's really difficult, if not impossible, to predict what the signaling aberrations are going to be just based on the genetic data alone. So sort of the answer to the first question is no. We really didn't go in with any assumptions of what the signaling would be. And I think that what we're finding is that there are many roads to Rome, and part of the heterogeneity that we see in AML is that there are so many different mutations and combinations of mutations that can lead you to the same signaling behavior. And that's what I find fascinating, is that all of these many different combinations of mutations are ultimately funneling into the same signaling behavior that mimics a normal stem cell. So your question about cell autonomous, I didn't quite catch that. What is your...
Well, I guess I was asking if there's no difference in surface receptors. Some tumors are, of course, requiring a growth factor in the environment. Mm-hmm. And then if there's no difference between that and a normal cell, and it's all signaling inside the cell from some mutation, then that may tell you one that's requiring a factor, estrogen or something. I don't know, whatever. I'm not a cancer guy.
That's a great idea, yeah. I mean, we did not measure every possible cytokine receptor on the outside of the cells. So we can't say for sure that the signaling behaviors we're observing are not driven by surface receptor expression. But I think that there are probably many different combinations of phosphatase mutations and kinase mutations and receptor overexpressions and things like that could lead you to these different phenotypes.
So sorry, because I don't mean to.
No,
just because you said something before, I just want to. So you're saying that the genetic data did not predict. You could not use the genetic data to tell you what the signaling defect would be based on the CyTOF?
Right. We only had very limited signaling genetic data on most of these samples.
But you did have some whole genome, and it wasn't able. That's interesting.
Right.
Wow.
Any other questions? Okay, well, thank you very much again to the organizers.